Hey guys, Professor Davis here, and we are going to be looking at the second part of energy for cells, and we're going to specifically talk about cellular respiration in this video. So a couple of things about cellular respiration. First of all, living cells cannot store a significant amount of free energy for their cells. And so because of this, we have to have a way that we can constantly make energy, okay? Because cells would get too hot, they would denature their own proteins, which then would in turn would cause problems for their enzymes, which are also proteins. And so this would create a kind of chain reaction of negative events for the cell. So cells constantly need energy in order to function. And the type of energy that they prefer is going to be an ATP. It's the safest form as well as the most efficient form of energy for the cell. Now, cells need to generate ATP, and they are going to do it through this process of cellular respiration that we're going to talk about today. Now, the way that cellular respiration kind of works is, first of all, glucose is going to be broken down through kind of a three-step process. First, we're going to see glycolysis take place. Then the products of glycolysis are going to then be modified and enter into the citric acid cycle, also known as the Krebs cycle. And then ultimately, some of those other byproducts are going to enter into the electron transport chain, which is going to be a type of oxidative phosphorylation. Now, you're probably sitting here thinking, man, that's a lot of words. I'm not real sure what they mean. Well, it's all right. We're going to go in and talk about them in this video. Cellular respiration is the conversion of carbohydrates and other metabolites like fats and proteins into ATP. Okay, now your cells prefer the carbohydrates. Your cells are kind of lazy. They prefer the carbohydrates because they're the easiest. However, your body could use fats and proteins for this process. Now, ATP provides just the right amount of energy for cellular work, and this helps with having little to no waste, okay, when we use ATP. Glucose is the most common molecule used to generate ATP, and the conversion of glucose can actually help make a large amount of ATP, which is about 39% efficient. It's one of the most efficient things of energy conversion we have out there, even more efficient than most machines. So what happens to the rest of the energy? We talked about this last video. Remember that when we transform energy from one type to another, it's going to be lost, not disappeared. It just means that it's going to be made into heat, which is not something that can be utilized by the cell. Okay, and so heat is what's generated and it cannot be utilized by the cell. So glucose is a type of, again, carbohydrate. We've already talked about that. And why is ATP used for cell work and not glucose? Well, ATP releases just the right amount of energy. You can kind of call it the Goldilocks effect. It's not too hot, not too cold. It's just right. And glucose actually releases too much energy at one time, so that's wasteful. But also, if we release too much energy at a time, it would cause potential combustion to occur, okay, fire, that sort of thing. And so we want to be able to break the glucose down in steps like you see here in this picture, and at each step we're going to release some ATP that's being formed. This is going to be a safer and more efficient way for the cell to gain access to that energy. Now remember, carbohydrates are the main thing we like to use. However, lipids or proteins can all do this, be converted into ATP through the process of cellular respiration. So guys, here's the chemical equation of cellular respiration. We start first with the sugar, so CH2O to the N. This is different types of sugars. And we also are going to need oxygen to be present. When we have oxygen present, we are able to convert this sugar into carbon dioxide and water, with ATP coming out as the energy source. Now, this particular molecule right here, when we talk about the idea of the carbohydrate, what's going to happen here is those H's of that carbohydrate are going to be lost. They're going to end up going away. So they're not really going to be lost. These hydrogens are going to be transferred. They're transferred from the carbohydrate to the water to create this H2O. Now, oxidation is a term used to indicate that a molecule has lost hydrogen atoms. Okay, and so because of this, if we're looking at this picture right here, which reactant's being oxidized during cellular respiration? Which reactant is losing hydrogens? Well, that would be the sugar. Okay, the CH2O, they lose the hydrogen, so they are going through oxidation. On the other hand, reduction is the term used to indicate that molecules have gained hydrogen atoms. And so what molecule of the reactants gained the hydrogens? That would be oxygen. So this is reduction. We reduced oxygen. Now guys, if you'll notice though, you have to have one of these in order to have the other. So a lot of times we call these oxidation reduction reactions or redox reactions, okay, because they always come together. Now this brings us back to some terms we saw from the previous video, endergonic and exergonic reactions. So if we have C6H12O6 and we are going to break it apart into CO2 and water, H2O, this is an exergonic reaction because we have energy that is being released. Now remember, this energy being released is going to be used to drive the next reaction, which is ADP, adding a phosphate to form ATP. Since we're adding something on here, this takes energy, which means it's an endergonic reaction. So we have an input. And so the energy that exits the first reaction drives the second reaction. So again, another concept here is exergonic reactions are going to always be coupled or put together with endergonic reactions. Now, this brings us to a few terms when we talk about oxygen. If oxygen is present and the process uses oxygen, this is an aerobic process, okay, because oxygen is there. If no oxygen is present, we call it anaerobic. Anytime you add AN to the front of a word, it means not or without. So in this case, it's without oxygen. So does the chemical equation following here represent an aerobic or anaerobic process? So I have C6H12O6 plus O2, which is oxygen, and we're going to convert this into CO2, H2O, and 36 ATP. So is this aerobic or anaerobic? In this particular case, this is aerobic because oxygen is present. On the other hand, does this chemical equation represent aerobic or anaerobic processes? So here we have C6H12O6. This gets converted into a C2H5OH plus carbon dioxide plus 2 ATP. If you look here, oxygen is nowhere to be found, so this is an anaerobic process. Another thing I want you to note about this is look at the amount of ATP made in the aerobic versus the anaerobic. In the aerobic respiration, we see 36 ATP were formed in this process, but in the anaerobic, there's only 2. Okay, oxygen is critical in order to make a large amount of ATP here. If oxygen is not present, you're not going to be able to make much ATP. Another thing too is that when we are able to create or make a lot of ATP, it's going to create low energy products like CO2 and water. Those are low energy products. Whereas on the other hand, in our second equation, C2H5OH still has a high energy level, and this is known as an alcohol. So another concept here is oxygen must be present for glucose to be completely oxidized into CO2. Okay, so oxygen has to be present in order for us to completely break down that sugar, that glucose, into carbon dioxide. So the first process up here where we have the oxygen present is known as aerobic cellular respiration, whereas the second equation is known as anaerobic fermentation. Now, what organelle inside the cell is important for this? This is the mitochondria. Now, the mitochondria, if you'll recall, is known as the powerhouse of the cell. This is going to be where the citric acid cycle or Krebs cycle takes place, as well as the electron transport chain. So the mitochondria is a very important organelle in this process of cellular respiration. So generation of ATP. How do we make this ATP? Well, I have to take ADP plus a phosphate. When I add that phosphate to ADP, it forms ATP. This is a process known as phosphorylation. Phosphorylation is just a big word that means I added a phosphate. Okay, so it's phosphorylation, I'm adding a phosphate. Now there's 2 types of phosphorylation we're going to discuss. The first one is substrate level phosphorylation, or SLP. This is where the phosphate group is removed from a substrate and added to ADP, and this is going to be done with the help of enzymes. So this is going to be done with the help of enzymes like you see in this particular picture. So in this case, the substrate comes into the enzyme, it's going to break off the phosphate that's present, and it's going to add that phosphate to ADP. This can only generate about 4 ATP because we get 2 from the glycolysis step and we get 2 from the Krebs cycle. So guys, the Krebs cycle and glycolysis use substrate level phosphorylation, or SLP. Another type of phosphorylation that's present is called oxidative phosphorylation, or OP. This is again the same process of adding a phosphate to ADP in order to create ATP. But guys, this occurs specifically in the cristae of the mitochondria, in those folds of the mitochondria. This type of phosphorylation, oxidative phosphorylation, is driven by hydrogen atoms. These hydrogen atoms are charged electrons in a proton, and they are being dropped off to the electron transport chain. So guys, the ETC here means electron transport chain. And they are being dropped off by 2 taxi type cabs, 2 what we call carrier molecules. This is NADH and FADH2. These are formed through the process of glycolysis, the prep stage, and the Krebs cycle, which we're going to talk about more in a minute. But they get to pick up hydrogens from those steps, and they're going to go and drop it off to this last step known as the ETC. Now, the ETC is coupled with something called chemiosmosis, and so that's the movement of chemicals, and ultimately water is going to be created in this process. This type of phosphorylation, guys, creates a lot more ATP than we saw with substrate level. This type of phosphorylation can actually generate anywhere between 32 and 34 ATPs. So NADH and FADH2 are going to drop off their hydrogens and their electrons, and these electrons are going to move down the electron transport chain. These electrons help generate the ATP, and it pumps the hydrogens into the inner membrane space between the outer membrane and the cristae. Okay, so they are going to pump them into this area here. These hydrogens then are going to go through an enzyme called ATP synthase. We know it's an enzyme because it ends in ase, and this particular enzyme is going to synthesize ATP. As the hydrogens move through here, this is known as chemiosmosis. This is going to turn this particular enzyme, allowing for ATP to be created. Ultimately, these hydrogens will get reunited with their electrons, and oxygen is going to be there to catch them, kind of like a parent at the bottom of a slide to catch their kid. They catch the electrons as well as the hydrogen ion, and this is going to be what creates water. Okay, so we have hydrogen atom plus oxygen is going to help create water, H2O. So let's talk about a quick overview of cellular respiration. We're going to go into a little more detail in each of these steps in just a minute, but substrate level phosphorylation occurs in some of them, as well as oxidative phosphorylation happens in other steps. So if you take a look here, you have glycolysis, it nets 2 ATP and it does substrate level phosphorylation. But if you'll notice coming off the top, it's going to create 2 NADHs. Those 2 NADHs are going to ultimately, if you trace the arrow, they're going to end up at the ETC, or the electron transport chain. We also have a prep stage that takes place inside the mitochondria. In this prep stage, two more NADHs are made, and they go also to the electron transport chain. And then you have the Krebs cycle. The Krebs cycle generates 2 more ATP. It also does substrate level phosphorylation. It also sends 6 NADHs as well as 2 FADH2s to the electron transport chain. This electron transport chain then is able to make 32 to 34 ATPs through oxidative phosphorylation. So which process generates the most ATP? Well, that would be oxidative phosphorylation, and more specifically, that would take place in the ETC, the electron transport chain. And this means that if oxygen is present during aerobic respiration, we ultimately could make anywhere between 36 and 38 ATP per molecule of glucose. Okay, so we can make 36 to 38 ATP for each molecule of glucose. So let's talk about the phases in aerobic cellular respiration. Now, if oxygen is present, glycolysis is followed by 3 kind of pathways. Glycolysis is going to take place no matter what because it takes place in the cytoplasm. But the next three steps are dependent on whether oxygen is present. So you have the preparatory stage, the Krebs cycle, also known as the citric acid cycle, and the electron transport chain. These three, oxygen must be present for the chemical reactions to occur inside the mitochondria. So if you look at the picture here, glycolysis happens out in the cytoplasm, but the other three steps, the prep stage, the Krebs cycle, and the electron transport chain, those take place inside the mitochondria. And you can think of it as oxygen being the ticket to get inside. If oxygen's not there, they cannot get inside of the mitochondria. So this reactant here is the C6H12O6. Okay, this is going to ultimately get converted into CO2. Now these hydrogen atoms, there's 12 of them, they slowly get stripped off glucose and they will get shuttled to the electron transport chain. The shuttles or the taxi cabs that do this are NADH and FADH2. Now, in the presence of oxygen, one molecule of glucose will yield approximately 36 to 38 ATP. Okay, 4 ATPs are generated through substrate level phosphorylation, and 32 to 34 are generated via oxidative phosphorylation. Hydrogen atoms are supplied by the glucose, the sugar, and these hydrogen atoms are carried to the electron transport chain of the mitochondria, the coenzymes NAD+, which become NADH, and FAD, which become FADH2. When NAD and FAD pick up their hydrogen atoms, they are reduced. Remember, if they accept the hydrogens, they become reduced. And remember that these are not just hydrogen carriers, they are also electron carriers. All right, so let's do a brief look at the steps of cellular respiration, and then we'll dive into them in a little more detail. So glycolysis, guys, takes place in the cytoplasm. Glucose is what goes into glycolysis and pyruvate comes out. This does occur in the cytoplasm. Oxygen is not needed and it's not required. So we're not going to require it. We're not going to use it. ATP, we're going to make 2 of them through substrate level phosphorylation. The prep stage is going to take that pyruvate from glycolysis and it's going to convert it to acetyl-CoA. This takes place in the mitochondrial matrix, okay, the fluid of the mitochondria. In this particular case, oxygen is required, but it is not used. So oxygen is required, but not used. In this step, no ATP is made. Now that acetyl-CoA is going to then enter into the Krebs cycle. When it enters into the Krebs cycle, it's going to merge with another product and it's going to be turned into oxaloacetate. This oxaloacetate is ultimately going to be a citrate plus that acetyl-CoA. So acetyl-CoA gets added to the citrate and it creates oxaloacetate. This also takes place in the mitochondrial matrix, so inside of that fluid and then in the mitochondria. Again, oxygen is required, but it is not used. In this step, we get 2 substrate level phosphorylation ATPs. So we get 2 ATPs here for the Krebs cycle. So far we have 4 ATPs from 1 glucose. Now, through these processes, glycolysis, the prep stage, and the Krebs cycle, hydrogens are constantly being broken off, and they are going to travel with the help of NAD and FAD to the electron transport chain. These hydrogens are going to get dropped off, and oxygen is going to be present to help make water. This takes place in the mitochondrial cristae, the folded part of the mitochondria. Here, oxygen is required and it is used. So oxygen is required and it is used. And this is the big step of ATP. We get 32 to 34 ATP, but we're going to get it through oxidative phosphorylation. So important here, guys, is aerobic cellular respiration includes glycolysis, but so does anaerobic. Anaerobic process means no oxygen is needed there. All right, so let's take a closer look. Now, if you look at this diagram here, there's a lot of stuff going on. There's a lot of boxes and a lot of different arrows and all. We're going to simplify this. Now, if you end up taking a more advanced type of biology class that focuses in on cellular respiration, you're going to need to know these steps in more detail. But here, we're just going to want a general breakdown, okay? That's the purpose here on this particular video. So we're going to first start with the process glycolysis, and we're going to start with a 6-carbon glucose, because remember, glucose is C6H12O6. These little dots represent the carbons. Now this glucose is going to get broken in half. That's what glycolysis means. Glyco means sugar, lysis means to break. So we are going to break this sugar in half. When this occurs, we're going to get 2 pyruvates. And pyruvate, guys, is a 3-carbon molecule, but we have 2 of them because what's half of 6? Half of 6 is 3. In this process, ATP is required to break this stuff. Okay, it takes energy to make energy here. So we're going to break this ATP. We're going to end up breaking this glucose. ATP goes out, but ATP comes out as well. So ATP goes in, ATP comes out. Also, NADH comes out. So what are our inputs and our outputs? Our inputs are glucose, and we're going to put in 2 ATP. Our outputs are pyruvate, and we have 2 of them. We also have ATP, which gives us 2 net. So 2 went in, but I made 4 because I got 2 left over. And we're going to have 2 NADHs. So what went into glycolysis and what came out of glycolysis, inputs and outputs. And remember, this process takes place in the cytoplasm. And if you'll notice too, oxygen does not have to be here. Oxygen has to be present here in order for us to enter into the actual mitochondria. And these pyruvates go from the cytoplasm into the mitochondria, and they're going to go into this preparatory stage, or this prep stage. So pyruvate goes in, and it's going to get converted into acetyl-CoA. So if you'll notice here, one of the carbons gets broken off, and the other 2 carbons get attached to CoA. So we have acetyl-CoA. This 1 carbon that broke off is going to merge with oxygen, and it's going to create carbon dioxide. We're also going to see an NADH formed here. So our input was pyruvate, and we had 2 of them go in because, right, there were 2 that left glycolysis. We're going to get 2 acetyl-CoAs. We're going to get 2 CO2s, and we're going to get 2 NADHs. Okay, so we're going to get 2 NADHs. Now, this is the prep stage. The prep stage does not make any ATP. Our next step is the Krebs cycle. Now, the Krebs cycle is going to turn twice for each glucose. So first of all, we're going to get acetyl-CoA to come in, and it's going to merge here with oxaloacetate. When this occurs, oxaloacetate has 4 plus 2 more carbons. We now go back to a 6-carbon molecule we call citrate. Citrate is then going to go through a series of events where we're going to see some things broken and moved around, and ultimately some NADH is made, some FADH2, ATP, and another carbon dioxide. Now this is with each turn. So input are 2 acetyl-CoAs, so this is going to turn twice, which means ultimately I'm going to end up getting 6 NADHs. I'm going to get 3 from the first turn and 3 from the second turn. I'm also going to get 2 FADH2s. I'm going to get 1 from the first term and then 2 from the second term. I'm also going to generate 2 ATPs, 1 from each turn, and finally CO2, carbon dioxide. Okay, so this is a Krebs cycle. What went in, what comes out. Last but not least, we have the electron transport chain. In the electron transport chain, these hydrogens are being dropped off by NADH and FADH2. ATP is ultimately going to be created. And this is due to the fact that hydrogens will move from low to high. They will then sneak back through this ATP synthase. This turns and creates ATP. Also, some of these hydrogens and their electrons are going to be accepted by oxygen. This is where we're going to use the oxygen. When we add hydrogens to oxygen, we end up with water. So what goes in to the electron transport chain? Well, 10 NADHs, and this is where the H comes from for hydrogen. We're also going to send 2 FADH2s, and these two are going to help produce the 32 to 34 ATP. We also see oxygen gets put in. Oxygen is going to accept some of these hydrogens and electrons in order to create H2O, which is water. So guys, this is a very complex process, but I tried to make it very simplified for you. You need to focus on what goes in and what comes out of each stage. So here's another overview. It shows you glycolysis happening outside of the mitochondria. The prep stage, the Krebs cycle, and the electron chain happens in the actual mitochondria. ATP gets made out of ATP gets made out of glycolysis by 2. We get 2 ATPs from the Krebs cycle, and you get 32 to 34 from the electron transport chain. This electron transport chain is able to do its job because it receives hydrogens and electrons from NADH and FADH2, which are made by each of the 3 previous stages or steps. Again, here's another pictorial breakdown, okay, where you can see we go from glucose to the prep stage, to the Krebs cycle, to the electron transport chain. And it shows you what goes in the sense of the main product, the main reactant, and it also shows you the amount of ATP that comes out of each step. So some interesting facts about ATP production. Cyanide, okay, which is a type of kind of poison, inhibits cytochrome C oxidase. Now this is an enzyme, okay, so we see oxidase tells you it's an enzyme, and it's a component of the electron transport chain. So if this messes up the electron transport chain, cyanide is going to actually cause your ATP to not get made properly, and so you're going to end up being deficient for energy. Some poisons inhibit not the oxidase enzyme, but they inhibit ATP synthase from working. Now, rigor mortis, which is when a dead body, when somebody dies and their body stiffens up, is due to the lack of ATP. If you want to take an anatomy talks about how with the muscles, there's energy that has to contract the muscle, okay, where it contracts, but you also have to have energy to relax. If you cannot replace that energy because the cells are dead, you're going to tighten up and all your muscles tighten and they cannot relax. After about 24 hours, these proteins do relax, but that's how come they can sometimes give you a time of death by looking at how much rigor the person was in. Mitochondrial disorders can also be caused by mutations in nuclear DNA, or they can be due to mutations in the mitochondrial DNA, because remember, the mitochondria has its own DNA. Another kind of interesting fact is some tissues have more mitochondria than others. This is what gives you the difference between light meat and dark meat in chickens. The dark meat is in the legs because there's a lot more myoglobin, like legs and thighs, because they have a lot more ATP because chickens walk, they do not fly. The breasts and the wings have the light, okay, or the white meat because they do not have near as many mitochondria, again, because they don't fly, they walk. Okay, so this is just giving you an interesting fact there about the light and dark meat in chickens. All right, so we talked about the process of aerobic respiration where oxygen is present. What happens when oxygen is not present? When oxygen is not present, pyruvate is the pivotal metabolite in cellular respiration. And whether oxygen there or not is going to determine what happens to that pyruvate, or that pyruvic acid. In man, the major driving force of this is oxygen. So if oxygen is present, pyruvate enters into the preparatory stage and aerobic cellular respiration occurs, like you see in this diagram. However, if oxygen is not present, fermentation occurs, oxygen does not get used, and water is not formed, like you see in this other side of the picture when oxygen is absent. So let's talk about what happens with fermentation. Fermentation is glycolysis. We're going to take that glucose and break it down into pyruvate, and we do see 2 ATP get made. Now in yeast cells, this process is going to be where the pyruvate is going to be kind of converted into CO2 as well as C2H5OH, which is an ethanol. Ethanol is an alcohol, and this is the product used for fermentation to create alcohol for us. On the other hand, in animal cells, pyruvate is going to get converted into C2H5COOH, which is lactic acid. Now, due to the oxygen debt, lactic acid is going to cause the negative or lower pH in your cells, and this is what causes your muscles to fatigue and get tired. Oxygen recovery is where some of the lactic acid is converted back to pyruvate in the liver, and it can be recycled. But guys, the lactic acid is responsible for making your muscles sore. And the reason being, guys, is ethanol and lactic acid are both products of fermentation, but they're both very toxic in their composition. Now, oxygen recovery can help us go backwards, but we've got to make sure enough oxygen is given to your cells. So what are the advantages and the disadvantages of this idea of anaerobic fermentation? Well, advantages are a rapid burst of energy. You can get energy really quick. You do not need oxygen for it, and most organisms can use this type of energy. But disadvantages include the fact that this is a short-lived process, can't last very long. It gives you a low amount of ATP, you only get 2, and the products are toxic. This also means it's a lot less efficient than aerobic respiration is. Now, there are some economic benefits to this, like when we use it for yeast, because yeast can help us make beer, wine, and bread through this process. And this anaerobic bacteria can also help us make cheese, yogurt, sauerkraut, and soy sauce. Okay, those are types of food, but they also can help us make isopropanol and acetic acid. Okay, and so there are some actual useful things that these processes of anaerobic fermentation can do for us. Now, guys, ultimately, remember, all energy comes from the sun. Photosynthesis is going to take place, which we're going to talk about next chapter, and it helps produce glucose. Glucose can now enter into the cells, and it can go through glycolysis. Glycolysis creates the pyruvate, and the pyruvate, depending on if oxygen is present or not, determines its fate. If oxygen is present, we call that aerobic cellular respiration. This gives us complete oxidation, and our waste products are water and carbon dioxide. And our net energy that we are able to then utilize is going to be 36 ATP per glucose. On the other hand, if we look at anaerobic, where oxygen is absent, this is called fermentation. This is incomplete oxidation of pyruvate. The waste products are things like lactic acid or ethanol, as well as carbon dioxide. And the net energy that we're able to then get for each glucose is only 2 ATP. So this summarizes the process of both cellular respiration and fermentation for you. Now, guys, what about if glucose is not present? Glucose has been talked about over and over for this, for glycolysis and that sort of thing. Well, guys, remember that metabolism is all of your chemical reactions. This includes the anabolism, which is the building of molecules, the synthetic part, and also the catabolism, which is the degradative or breaking down part. So anabolism builds things, whereas catabolism breaks them down. Catabolic reactions release or produce energy, which then are going to be used to drive anabolic reactions. We have to break down our food, releasing energy in order for us to build other things. Now, other types of molecules can be used as metabolites besides just glucose to make ATP. This does include proteins and fats. But guys, these pathways are not closed systems, and so because of this, there are some differences that have to take place. So fructose and galactose are other types of sugars besides glucose that produce the same number of ATP as glucose. However, sucrose, which is a more complex sugar, it's a disaccharide, and lactose can be broken down into their monosaccharides and then enter the pathway. But there's an extra step that has to happen to break those down. Amino acids must have the amino group removed and convert it to ammonia to enter the pathway. So proteins can also enter this process, but it takes an extra step. And then glycerol can be phosphorylated and processed through glycolysis, as well as fatty acids can be broken down in order to enter the citric acid cycle or the Krebs cycle. So we do see that we can utilize other things, but more steps are required. As this is one reason why some diets will have you cut out carbs. If you cut out the carbohydrates and you make your body use proteins and fats, it's going to use more energy to get that energy. All right. And so because of that, it could help you potentially lose the weight because you're having to break into your reserves, okay, your fat reserves or even your protein. So when we look at proteins, guys, one thing I want to hit on is that when we talk about proteins and amino acids, there are 20 of them. 11 of them are non-essential, meaning that you can get them by making them. You can make them yourself. Your cells can put them together. But 9 are essential, and you must get them from your diet because you cannot make them. So where do you get these proteins from your diet? Well, you're going to get them through eggs, dairy products. You can do it through poultry or other seafood. And they provide all 9 of the essential amino acids as well as other meat products as well. Eggs are a really good example of being able to get all of those. So these are just showing you that there are some non-essential, meaning that you can make them, but there are 9 essential amino acids that you have to get from your diet. It's the only way you can get them. All right, the last thing I want to talk to you a little bit about is exercise. Okay, so when you exercise, you're going to need a lot of ATP for your muscles to function. But also on top of that, you need to make sure you're giving your cells oxygen so they can replace that ATP. Prolonged aerobic exercise can ultimately burn fat though, because you're not constantly eating carbs while you're working out, or at least I hope you're not. And so because of this, burning off fat requires oxygen to be there. You cannot do anaerobic respiration and burn your fat. It has to be aerobic. If oxygen runs out through vigorous exercise, like you're sucking air, because you're not getting enough oxygen into your bodies, cells will switch to fermentation. And fat burning decreases. And so guys, when we talk about people getting in shape, it's not so much about this kind of shape. It's talking about getting shape in your lungs and your heart so it's more efficient at delivering oxygen to your cells. The better they are able to get oxygen to your cells, the better your cells are able to break down fat. So if you want to lose like a few extra pounds, you will have to have more success with aerobic exercise than anaerobic exercise. Brisk walking, biking, swimming, and jogging could be beneficial for that type of exercise. Other benefits, though, of aerobic exercise, besides just helping burn fat, is it helps your heart and lungs get stronger, more O2 to your muscles, and again, they become more efficient. Skeletal muscles blood supply increases and the number of mitochondria also increase, again, making sure your reserve of ATP is there. And your muscles can use more energy even when your body is at rest. We find that the more muscle mass you have, the more you burn calorie-wise while you're just resting, sitting, and even sleeping compared to if you don't have that muscle mass. So the longer you exercise, the more likely you are to burn fatty acids. However, this is difficult. We can't say you can exercise for 5 hours at a time. So you need to really make the best of the exercise you do. Another thing too is that it's not just exercise alone. Your diet also may need to be addressed. In anaerobic exercise, you can develop stronger muscles through that process. It does improve your ability to consume oxygen during exercise as well by improving your cardiovascular fitness and increasing your capacity to withstand the buildup of waste substances like lactic acid, and removing it from your body is super important. This is why if you've ever had a personal trainer or a coach or something like that, when you've been exercising, even when you're lifting weights where you're not just doing like running or jogging or biking, they want you to breathe while you are exercising, because that breathing process is going to supply oxygen to your muscles. It's going to help you not be as sore. It's also going to help with your fitness with your hearts and lungs, and it helps build your muscles better as well. All right, so this finishes up the idea of cellular respiration. If you have any questions or concerns, please let me know.